Look, so while we wait for this, so I'm going to talk about some connectivity. This is in a rodent and using a mouse model. And, the connectomics that's coming out of the human and primate literature is fantastic. But in terms of assigning functional roles for these circuits, it's a lot of it comes in the humans studies from psychiatric disorders, and we're going to hear this afternoon in a little while after about from DBS type studies. And this is assigning, looking for functional roles. Now, in the primate system, of course, people, in the visual system, the nervous system, have been working for many, many years. And as most of you in this room would know, it comes with its own set of issues, or the kinds of things that are possible to do. And the rodent system, of course, offers a lot of advantages in this space here, where you have the ability to manipulate and carry out, I guess, both lesions as well as using, things like optogenetics. And the advantage really of the rodent system is speed more than anything. And often a lot of the tools have been worked out there, and it's slowly moving to the primate world. But I guess in many ways, outside, at least in Australia, working on primates is becoming very, very difficult, and there's all sorts of ethical issues associated with it. So the mixture between the rodent side is, I think there's a lot of people really strongly believe that the functional roles probably will get sorted out on the rodent side first, and then applied maybe on the primate side. And this is the space that, this connectome project is really working towards and bringing together both sides of the story. So I'll give you the story that I'll tell you is interactions between the ventral hippocampus, prefrontal cortex, and the amygdala, and trying to think about, how do you attack the idea of how is it a circuit performs a particular behavioral outcome. So this works. Thanks very much. Look, so the paradigm we've been using is one that's used by so many different people around the world. This is fear conditioning. And, fear conditioning is pretty simple. It's Pavlovian, and you pair a neutral stimulus. Typically, we use a tone, or you could use a sound, or you could use a light, or a smell, doesn't really matter. And in Pavlovian conditioning, there's an acquisition phase in which a conditioned stimulus, which is neutral, is paired with the aversive ones, or it could be food. It doesn't really matter which direction you go. You pretty much get the same result, in which the animals learn to associate the two, and they learn, and this is the acquisition phase. Now, you come back, animals learn to associate the two, and you can come back a month later or a year later. In humans, you can come back many years later, and you test with the same conditioned stimulus, and they respond with pretty much the same. So there's a memory formed of this learning process. Now, there's also a process called extinction, in which if the conditioned stimulus is now paid repetitively without pairing it with a foot shock or with a reward, you get this phase called extinction, in which the animal now learns that this stimulus is no longer aversive. And this has been known since the time of Pavlov, that this is not a removal of the old memory. It's just new learning, in which you learn that this previously a reward producing or aversive stimulus is now no longer scary or no longer rewarding, and you learn to respond not to it. Okay, so this is extinction, and extinction is pretty much what's used in aversive therapy in conditions like post-traumatic stress, in phobias, and in drug addiction for that matter. What's interesting about extinction is that extinction is context dependent, and that is that if you move from the context in which you learned this was no longer scary, so this is like a different context now. So for instance, you could be extinguished in this room or lose your addictions in this environment. You move to a different environment, and there it all comes back. The whole behavior all comes back. So it's context dependent, and this is what's called relapse. Okay, this is what's relapse, and this is part of the problem with extinction therapy. So we're pretty interested in trying to understand how is it that this relapse is happening. Now, the circuitry that underpins fear conditioning has been worked out over many, many years, and it's pretty clear it involves the amygdala. And I'm not really going to go through the exact circuitry for fear learning, but what's very clear is that the medial prefrontal cortex and what's been commonly called prelimbic middle prefrontal cortex, is involved in fear expression. That is, at the time that the learned response is again produced, that is when you play the tone and the animal freezes, or you play the tone, animal learns to find reward, the prelimbic cortex, this bit of the brain, which is a 4-layered cortex rather than a 6-layered cortex, projects to the amygdala and is involved in expression. Now it turns out that in extinction what happens is that while the prelimbic part of it is still active, the infralimbic part of it comes into play, and this is what causes extinction. So what's commonly thought is what happens is that projections from the IL to the amygdala, in fact what they do is they turn this fear off basically. Now extinction is context dependent, as I just showed you. Okay? And what's very clear is that this extinction is driven by the ventral hippocampus. And the ventral hippocampus kind of makes sense. The ventral hippocampus is an area of the brain which is involved in learning about where it is you're in space. Okay? So what we're interested in trying to understand is, how is it that this circuitry operates? What is exactly going on? Okay, so here's the evidence that the IL, the infralimbic part of the medial prefrontal cortex, is in fact involved in driving extinction. This is a very simple behavioral paradigm. Animals are fear conditioned in a box, and then they're extinguished in the same box. This is the orange box there. Okay? And in the same box, they're tested for extinction. Okay? And now what we do is we inject muscimol, which is a GABA A receptor agonist, into the IL and turn the IL off. Okay? So what it does is it activates GABA receptors, switches the thing off, and what you find is that fear, as you can see in this last panel here, fear comes back when the muscimol. This is actually not working, as far as I can tell. Right. So this is it here, right? This is your fear coming back. So this is extinguished fear and the fear coming back. Okay. So here's showing the renewal effect, if you like. Okay, now we're trying to look to see what happens in renewal. So what's happening here is that animals get fear conditioned in one environment, they're extinguished in the very same environment, called it the orange one, and then they're tested in the same environment where they were extinguished and a different one. This is shifting context. Okay. Now to look at the effects of this circuitry, what we've done is it's a dual viral approach. And this is the kind of thing that you can just about do in primates now as well. So you inject one virus into the IL, the infralimbic prefrontal cortex. This is a virus that's retrogradely transported and it expresses Cre recombinase. Okay, so it's retrogradely transported. So the cells in the ventral hippocampus that project to the IL are now labeled with Cre recombinase. Okay, then you come along with a different virus which has got a floxed construct on it, and you inject that into the ventral hippocampus. So now cells in the ventral hippocampus are now transducing your virus, and you're unfloxing whatever you're putting in there. So what you've done now, you've expressed a construct into cells which are going from the ventral hippocampus to the IL. So what we've done is we injected two types of constructs in there, and I'll show you two types of experiments. One is a DREAD, which is a chemically activated ion channel, either excitatory or inhibitory, and this is activated by CNO, by injection into as an IP injection, or we use channel rhodopsin, then we cut slices and look at the circuitry. So now what this lets you do, of course, is to do two separate things. Firstly, you can look at the behavioral impact of these different connections. You can also try to work out what the actual circuits are that underpin it. Okay? So here's now using a DREAD, and there's two separate DREADs. So in green is just a GFP control. Okay, this is just using a virus expressing GFP. The blue ones is an excitatory virus. You give CNO, it turns these cells on. And the red ones is an inhibitory virus. This is opening a channel which maybe hyperpolarizes the cells and turns them off. Okay, so all these animals are fear conditioned, and you see this show fear, and they're tested. This is the initial extinction. They're freezing, and they're showing fear. You extinguish them, and then the fear goes away. Okay. Now these animals are moved to a different environment, and you can see this is renewal. Okay, so these animals are freezing. They've been extinguished. With the GFP animals, they just freeze. Okay, this is the controls, and this is giving CNO, which activates the DREAD. Okay, here's the animals in which the virus is expressing an inhibitory DREAD, And these, don't forget, these are the cells that are projecting from the ventral hippocampus to the IL. You turn these guys off, in the different environment. These animals which were freezing because of renewal, the freezing goes down. So now they're behaving as though the renewal didn't take place. You can turn the renewal off by turning the ventral hippocampal projections to the IL. Okay, so these are specifically turning off. Now you're not turning off the entire IL, you're simply turning off the drive from the ventral hippocampus, and we know this is glutamatergic. Now here's the opposite experiment, where the animals are tested in the environment where they were extinguished to begin with, and this is an excitatory dead now. You're turning on connections from the ventral hippocampus to the IL, and what you find is these animals are showing renewal in the context in which they were extinguished. Okay, so what this tells you is that these projections from the ventral hippocampus to the IL are driving the renewal. Okay, so whatever is happening during renewal, these projections are turning on, and that's what's driving the renewal. Okay, so what we wanted to ask was, so what on earth is this projection doing? What is going on? Okay, so what we did now is we simply injected channel rhodopsin into the ventral hippocampus. Now these experiments done with an injection directly into the ventral hippocampus. You can also do it with the dual viral approach. And now you go and cut slices and you record from cells in the IL and try and figure out what this projection is doing. Now, once we do the electrophysiology, we fix the slices and we reconstruct the anatomy as well. Okay, so if we record from cells, now these are pyramidal neurons. Now what's known about the IL is the layer 2-3 cells in the IL project to the amygdala, and this has been characterized in detail over many, many years. Cells in layer 5 also project, but it's a much smaller projection. So this is the characteristic firing properties of the cells in layer 2-3. You activate the ventral hippocampal input, and what's interesting is what you find is that this is an excitatory input. This is voltage clamp, and this is current clamp now, and you get an EPSB, an excitation. But what's dominant is the IPSB. You have a very long inhibition of these cells. And this, you can see, this is the inhibitory potential. In fact, it shunts the EPSB. So the projection from the ventral hippocampus to the projection cells in the IL is basically causing a very large inhibition. Now, if you record from interneurons in the IL, so this is now a fast spiking interneuron. Now we know these are cells express paraalbumin, so we know the kinds of interneurons that exist there. These cells get a very large input from the ventral hippocampus. These are driven to threshold, they fire action potentials. So the idea is that these cells, what the ventral hippocampus is doing is driving interneurons, and I'll show you evidence to say which interneurons these are. And what they do is they cause feed-forward inhibition and turn off the cells in the IL. Okay? So this is just quantifying this data now, showing cells in layer 2-3, cells in layer 5-6, and the interneurons. Okay? Just showing that the input to interneurons is tons bigger, basically. And in fact, the projection from the ventral hippocampus to the IL is very large compared to the projection to the prelimbic cortex. The prelimbic, don't forget, is the one that's causing the fear response to happen in the first place. Okay, so these cells that are getting turned off by the ventral hippocampus input, do they in fact project to the amygdala? So this is another kind of experiment you can do so easily in rodents. You take retrograde beads, you inject that into the amygdala. So now you can back-label all your cells in the IL, which are projecting to the amygdala. You inject channel rhodopsin into the ventral hippocampus. So now you've got projections from the ventral hippocampus going to the IL. You go and make recordings from the IL and look to see what happens to cells projecting to the amygdala. Okay? And in fact, this is one of these cells in layer 2-3. And you can look at this when we were doing this blindly. This is precisely what we saw in those cells. So cells which are projecting to the amygdala, and this is just quantifying the two types of cells. These are control cells picked at random. These are cells which we know project to the amygdala. They get large inhibition from the ventral hippocampus. So now we know that what the ventral hippocampal projection is doing is turning off cells which project to the amygdala. Now these cells are presumably what's driving the extinction behavior. The animals, the reasons that don't freeze in this environment where these cells turn on, and what that does is blocks the freezing. Okay? And the ventral hippocampus is turning that input off. So what's interesting about this inhibition is you can see it's very long time code. This is 100 milliseconds here. So this inhibition is lasting almost a second long for a single stimulus of the ventral hippocampus. This inhibition is blocked by this compound called CGP55845. And what this does is blocks GABA B receptors. This is the long-lasting inhibition, and you find it's very potent in the IL. Okay, this thing, so here we're showing that this input, when it's on, can actually turn these cells off. So here's where driving one of these cells that projects to the amygdala, you activate the ventral hippocampus, you cause this long-lasting inhibition, you completely stop the cell firing. So in fact, what we've shown here is that the ventral hippocampal input is so strong that it'll actually stop the cells in the IL spiking. Okay? So this is what's happening now. Here's the ventral hippocampus projecting to the IL. The IL, we know, projects to the amygdala. Okay? What this circuit does, we understand that as well. It projects to a set of cells called the intercalated cells. These are feed-forward interneurons to the central amygdala, and we can talk about this later if we want to. The central amygdala, it was causes all the output from the amygdala. These are the ones that are causing you to freeze or run if you're trying to escape fear from space. What the ITC cells do is turn this output off, okay? So basically, it's inhibition, inhibition, inhibition is what's coming from here. So this inhibition is turning off the output of the central. The ventral hippocampus is turning on an interneuron population in the ventral hippocampus, and it's turning off this output. Okay? So inhibition is working in two separate places, and that's how this is working. So we know that in the IL, there's at least two major types of interneurons. There's the pyrabmin interneurons and the somatostatin interneurons. And we know that the somatostatin ones make synapses on the dendritic tree. The PV ones are making synapses on the somas. Okay? There's also a third population, but they're much more tinier. So which cell is it that's being activated here? So now there's Cre lines available. This is the PV Cre line. These are animals who express Cre recombinase in pyrabrin interneurons. Use a flux virus into the IL, and what it's showing you is that indeed the PV interneurons cause the same sort of inhibition on the layer 2-3 cells as the ventral hippocampus did. Okay, so the PV cells can cause this kind of inhibition. This is the characteristic. Remember, this is the firing property I showed you exactly when we were making recordings from those animals with channel rhodopsin in the ventral hippocampus. Here's the somatostatin cells. All right, the somatostatin cells have a different firing pattern. These are called stutterers. These cells make synapses on the dendritic tree. They also cause inhibition. So both the PVs and the somatostatin cells are in fact connected to the pyramidal cells and cause inhibition. Very similar to what we saw from the ventral hippocampus. Okay? So which cell is it that the ventral hippocampus is turning on? So now we took a completely different approach here. So this is now a receptor called the ivermectin receptor. This has been engineered. This is a human glycine receptor. This is engineered to put into a mouse, and it reacts to a compound called ivermectin. And ivermectin is an antihelminthic, which is used in feed to rid animals of bugs, basically. So what we're doing here is we put ivermectin receptor into the PV interneurons, channel rhodopsin into the ventral hippocampus, and here's the stimulus of the ventral hippocampal afferents, and you get this large IPSP. You put ivermectin to silence the PV cells. What we've done now is turned off the PV interneurons, and this inhibition goes away. The ventral hippocampus goes away. Okay? So that's one approach. He's using a chemical approach to turn these guys off. So he's using a different approach now. This is using arch rhodopsin. This is now a chloride channel, which is turned on by light of a different wavelength. And we're putting these into the PV Cre animals again. And again, you can show this is turning off the PV cells, and again blocking this inhibition that comes from the ventral hippocampus. He's doing the same experiment using somatostatin Cre animals. So these animals, the Cre recombinase is just in the somatostatin interneurons. You can turn these guys off as well. Has no impact on the ventral hippocampal driven inhibition. Okay, so what's very clear is that the ventral hippocampus, it innervates a specific interneuron population in the IL, the PV interneurons. These cells selectively turn off the layer 2-3 cells. The SOM cells are also causing feed-forward inhibition, but presumably this is involved in a completely different circuit. Okay, so does it work behaviorally? So these experiments suggest that inhibition, which is in the IL, must be important for renewal. So here's another behavior experiment now. Animals are fear conditioned. They're extinguished in context A. They're moved into a different context where there's a renewal. So what we're doing now is simply injecting inhibitors of inhibition into the IL. Okay? So here's vehicle. These animals are showing fear. They're showing renewal. If you inject picrotoxin or CGP, you can basically turn off this renewal. Okay? The renewal just goes away. So this is evidence that the renewal, in fact, is caused by inhibition in the IL. Okay, so here's the summary. So extinction is mediated by cells in the prefrontal cortex which project to the amygdala and cause feed-forward inhibition of the central amygdala. The ventral hippocampus, and I haven't shown you this data, but the ventral hippocampus is turned on when you shift context. These cells are turned on. What they do is they project to somatostatin to paravalanol into neurons. These interneurons cause feed-forward inhibition of the layer 2-3 cells which project to the amygdala. So extinction is using inhibition, and now we understand the circuits that underpin this. So one interesting thing that arises out of all this is if you're thinking of renewal, and this might be something you might think about if you're trying to treat post-traumatic stress or even addiction-type disorders where renewal has major impacts, you might want to target this circuit between the PV interneurons and the layer 2-3 cells. Now, understanding these receptors here is pretty straightforward. You can work out exactly which receptors there are there, and then you can try and target this system to cure some of these disorders. Okay, so thanks for your attention. I'll thank the people who did the experiments here. So Roger Marek and Cornelia Strobel both did PhDs in the lab, and they both did postdocs and were involved in that local circuitry from the ventral hippocampus to the prefrontal cortex. Helen Gooch did all the channel rhodopsin experiments, and Li Zhu is our person who makes all our viruses. And the behavioral experiments were done in collaboration with Steve Moran and Gigi back at Dallas A&M. And Joseph Lynch and Tim Liner made the ivermectin receptors. So thanks for your attention.
Ivan Pavlov would be proud of you. Now, the pre-limbic and infralimbic, are they limbic?
They have, well, then, when you say they're limbic in the sense they're part of what used to be called the limbic system, I guess. They're sort of odd words now, which are probably going to lose currency in the future. They're actually, what's called the prelimbic cortex, if you look at the rostral parts of it compared to the caudal part, they're completely different. And in this literature, it's all seen as all one, basically. And as some of the parcellation studies are showing us, and we've done a lot of anatomy now, they get very different connections, the caudal and the rostral parts of it. And there's a group, Greg Quirk in Puerto Rico, who's also showing that functionally they have very different roles. And the IL will be similar.
Yeah. Now, I have a suggestion to make. The terms prelimbic and infralimbic clearly state that they are not limbic. And it is cruel to the students who come to the field because they throw them totally out. And I suggest something that will not only assist with this, but also make you more accessible to those who work with the human, to use instead 832 and 25. So
no, look, I agree with you, George. I tell you what the problem is, right? The problem is, like, the people who work in the field of fear conditioning and extinction, right? These words prelimbic and infralimbic are so buried in. I think what we'll have to do is you publish this stuff, and then you write a review suggesting that it's about time we changed it. And in fact, like the Gordon Conference for the amygdala, which runs every 2 years, this is where this needs to get brought up when the whole community is there to say, look, it's time we changed our nomenclature here, right? And certainly it's happening in the amygdala already, right? And don't forget, this is an area which has, been, hundreds of people have been working in it for many, many years. This circuitry here is just taking traction now, but it will change. There's no doubt it will change. Yeah.
Okay, any more question? Is there
Question here?
yeah.
Oh yeah, hi.
One more work. So I have two questions. The first is, I'm thinking the extinction and fear acquisition is kind of a continuous process, sort of like a lifetime, right? So I'm wondering whether during the natural extinction there will be a LTD, like from ventral hippocampus to IL.
Well, look, we haven't looked, right, but right now, from the single unit data that's coming from Steve Moran's lab, what it looks like is that the inhibition, let's talk about the renewal part of it first. The renewal part of it, seems to be driven by some other input that's coming into the driving those cells in the ventral hippocampus, right? And it's not really an LTP type mechanism. Now, the extinction part of it, the original part of it, that clearly requires some plasticity, again in the middle prefrontal cortex, because if you put in inhibitors of those things, they do interfere with it, okay? That plasticity, commonly it's thought to be LTP, not LTD.
But then related, but maybe opposite side of the question would be another possibility is will be a reorganization input. Right? So either an organization from ventral hippocampus to meaning because there's sparse coding of hippocampus for most of the context information. So if you imagine that this sparse input to sparse kind of IL neurons, whether it be side neurons, will produce kind of sparse kind of replication. And so there's plenty of room for the reorganization.
Oh, absolutely, absolutely. Look, I mean, and as you know, those are pretty difficult experiments, right? In the sense, and we've started going down that track by putting single unit recorders in the prelimbic cortex, as well as the hippocampus, as well as, and tracking all three places basically. And as you say, it is very sparse, right? And and this is the biggest problem with this technique, optogenetics. We're driving these circuits by bulk driving them, right, optically. And even with the dreads, we're silencing large populations of cells pretty much en masse, right, which is something that's never happening in vivo. So exactly how it operates with the sparse coding is something that will get sorted out, and it may well be that some of these circuits are plastic, yeah.
So now we stop here. So we have a group photo just here. So everyone welcome. The speakers maybe sit on chair. The first row. Okay, thank you.